Hello, here is a problem, a calculation, and I would like you to attempt at answering it. Assume 100 DATPs have polymerized into a single-stranded polyadenylate strand. What would be the molecular weight of such a strand? Given that we have the molecular weight of the DATP. So to answer this, we'll have to learn about the chemistry of polymerization itself. How nucleotides will polymerize and form a polynucleotide. To understand that, we need to understand something else, and more so because we need to understand that there are some components that we need to understand. Quickly, if we can try to recollect the numbering of the bases, I hope you can recollect that 1, 2, the nitrogenous bases are mentioned something like this, 7, 8 and 9. Here, this is 6 aminopurine, which must be adenine. And then we have 1 prime, 2 prime, 3 prime, 4 prime and 5 prime. That is, it is 2 prime deoxy, so this is deoxyribose. And 2 points, the polarity of DNA is referred to by the groups that are free on the phosphate ribose backbone. That is 3 prime OH. That becomes very important for us, these two. Similarly, we also have 5 prime OH, that is this 5 prime phosphate, because on the phosphate groups are attached to the 5 prime carbon. That is something also essential for the same reason. Then we need to also label them. This is alpha phosphate and beta and gamma phosphates. So how does polymerization occur? I am Dr. B.C. Amar, Assistant Professor at Shastra University. So for DNA synthesis to happen, and for the whole reaction in a way is something like this, where we have DNTPs and we make it into a polynucleotide, that is DNA, say DNA of a certain length, number of bases, what I meant by it. The number of bases given would form the DNA, and it is catalyzed by DNA polymerase. We write it DNA pol. And DNA polymerase is special, or the majority of the polymerases that we find in molecular biology that we read, they are all sequence dependent, because it is about the transfer of the information or physical manifestation of sequence information. So the sequence should be carried or should be read properly, and that is where majority of the polymerases, including if we count the DNA polymerases, RNA polymerases and also the ribosome, they all are read. They all read a sequence from a template. They are all template dependent, because it's not just polymerizing DNTPs to DNA, it is also putting them in proper order, and that is why they are template dependent. And there is one more thing that the DNA polymerase has. It's a kind of deficiency in a way, but a required deficiency, that it needs a primer, meaning that DNA polymerase requires, as we said, a template, and it also requires a primer, which we'll designate it this way. So primer is something which has a base, a nucleotide or an end of a polynucleotide with a free 3 prime OH. And so therefore I should put this is 5 prime, and this is 3 prime. OH is here, and this should be then 3 prime, and here is the 5 prime. So DNA polymerase will bind here to this place, this region is called as the primer template junction, and this is essential. DNA polymerase, all it can do is add more and more bases based on the sequence that is present here. So that is one of the reasons why primer template junction is essential for DNA polymerase to act upon. It is template dependent. So during this, at this point we are only looking at the chemistry of how things happen, and the rest, the enzymology and how actual replication happens, will be in the following videos. Here is a DNA polymerase. Here we have a nucleotide, and the DNA polymerase is occupying this, and what it does is that it binds to the, or it activates the alpha phosphate. It will allow the formation of this 3' OH binding to the phosphate group, alpha phosphate group, and these two phosphates, that is beta and gamma phosphates, are released as pyrophosphate. Pyrophosphate is further degraded by pyrophosphatase to release more energy, which drives the reaction of the polymerization itself, and that is how we have here a new base is added. For the stabilization of the incoming nucleotide, the nucleotide that should be added, the DNA polymerases will we have two metal ions, metal ion A and metal ion B. Here is the incoming nucleotide. It is given as this R1, and then we have the alpha, beta and gamma phosphates, and here is the R2, or that is the 3' OH. The 3' OH gets activated by losing one of the hydrogens, because of which the oxy anion is formed, and the oxy anion will make a nucleophilic attack with the phosphate and form a bond with the phosphate. It makes a phosphodiester bond and releasing the pyrophosphate, and thereby now we have one base added to the growing chain of the primer. And which base nucleotide should come here will be dependent on the template, which we will discuss later on. So if we want to solve this question, here we are saying 100 DATP molecules have polymerized into a single stranded polyadenylate strand, and then we need to, the molecular weight of the DATP is given, and we can figure out the molecular weight of phosphate groups, approximately around 94 grams per mole. And all during polymerization, what is happening is there is a loss of this beta and gamma phosphates, right? And alpha is retained. So if we take the first base, first nucleotide, say there is a DATP, and then there is a 3' OH to it, and to the 3' OH a phosphate group and the next DATP would have been added, to which OH would have been there, the 3' OH, and there would be a phosphate group added by the next DATP. Like this we can go 100 times, because we have given were about 100 ATP molecules, DATP molecules. And so in essence, to solve this question, all we need to consider is the first DATP would be in triphosphate. There would have been on the 5' side, we would have had 3 phosphate groups, which wouldn't have been lost. And for every other subsequent DATP, there would have been released of pyrophosphate groups, multiple of them. So in essence, if you are trying to solve this question, we could have taken 491, that is full ATP, we will just say it as, into 1 plus 99 into whatever molecular weight you would get of DATP. I would like you to solve this and post it in the comment. Thank you very much.